Dear students, welcome to Time class. So I am welcoming you all to the biology session. And in the last session, we have discussed about the concept of a cell. That is, cell is the basic unit of life. So it was our topic, the cell, basic unit of life, from the very first chapter, that is cell tissues and body movements. So in this session, we are not going to discuss the basics. Here you will get an exercise session. Exercise session means in the last session we have discussed some concepts. Based on that, there will be 4 types of questions. That is, there are some questions to test your basics. It will be direct questions in the form of objective type questions or fill-up questions to test whether you have studied the basics. And second type of questions will be, it's a knowledge based questions. It will be based on some concepts, some questions, descriptive questions will be there. Another category of question is understanding level of questions. How much you have understand, that is the concepts that we have discussed. And lastly, you will be having an application level of questions. These are 4 types of questions that we are going to discuss in this session. So before going to the exercise, you have to be thorough with your the topics that we have discussed in the previous. What are the things that we have discussed? Just point out. I'm going to just point out, and further discussion will be done while discussing the question and answers. Okay. So we have started with the discovery of a cell. Who discovered cell? It was Robert Hooke, in the cork slice, and he called it as cell, which means a little room. And further contributions of different scientists about Purkinje, Anton van Leeuwenhoek, and followed by the cell theory, followed by Schleiden and Schwamm, and it was modified by Rudolf Virchow, and there are 3 postulates about cell theory. Okay. Then next we have categorized the organisms into unicellular and multicellular based upon the number of cells they possess. Then we have seen the size of a cell and shape of a cell, and the shape of the cell may vary depending upon the function they possess. And again, organisms are classified into prokaryote and eukaryote on the basis of a true nucleus. Prokaryotes, that they have a nucleoid instead of a true nucleus. But in the case of eukaryote, they will have a true nucleus with a membrane-bound cell organelles. Then what is cytoplasm and what is protoplasm and what is cell wall and what is cell membrane, like that things we have discussed. Then we have seen the differences between prokaryotic and eukaryotic organisms. And after that, the structures of cell membrane, cell wall, and what are inclusion bodies, what are mesosomes, like that things we have discussed. And towards the last, we have discussed about cell organelles. What are cell organelles? And there are 2 types of cell organelles, that is membrane bound and non-membrane bound. And it is ribosomes, which is non-membrane bound, which is common in both prokaryotic and eukaryotic organisms. Okay. Then what is endoplasmic reticulum? What is their function? And what are 2 types of endoplasmic reticulum? That is RER, rough endoplasmic reticulum, and smooth endoplasmic reticulum, based upon the presence of ribosomes. And what are ribosomes? Where they can be seen? And what is the function of ribosomes? And towards the end, we have discussed the cell organelle, Golgi apparatus or Golgi bodies, about Camillo Golgi, who discovered, and what is the function, storage, packaging, and dispatch of the materials was the function of the Golgi apparatus. So with the Golgi apparatus, we have ended up last session. So in this session, based on that session, we are going to do some exercises, some practice exercise. Okay, are you ready? It is very interesting and it is very easy for you. It is not at all tough. So, moving on to discussion. Here comes our topic. Topic means the name of our chapter. Cells, tissues and body movements. And cell, the basic unit of life, is the subtopic name, is the subtopic that we are discussing, isn't it? So in every slide you can see this heading, and it is a group of questions that is to test your basics. That is, it is probably the direct questions from the topics that we have discussed in the last session. So directions for questions from 1 to 10 is fill in the blanks, fill up the blanks. Then the first question, the first living cell was discovered by. So please read the question carefully. The question is like this. The first living cell was discovered by. It was not asked the cell was discovered by. The living cell was discovered by. So what all things do we know? That is, the cell was discovered by whom? Robert Hooke. But Robert Hooke, he couldn't find any living substances like cytoplasm or nucleus or any other organelles inside the cell. It was Anton van Leeuwenhoek who discovered the living cell, that is a cell with a cytoplasm and nucleus, where he found that Robert Hooke found the discovered cell in a cork, that is a thin slice of the bark of a tree, through a self-defined microscope or primitive microscope which was designed by him. But it was Anton Leeuwenhoek who discovered living cell, first living cell in pond water. The water, okay, pond water in pond, in a algae known as Spirogyra. About algae and all you will be studying in the higher classes. Okay. So first living cell was discovered by whom? Anton van Leeuwenhoek. Okay, yes. So I know you know the answer, but I can't hear you because it is online session. Okay, so let us check whether the answer is correct or not. Yes, Anton van Leeuwenhoek is the correct answer. Then next, dash is the basic structural and functional unit of life. So you know it is a very easy and very direct. The name of our subtopic is itself. It is the basic structural and functional unit of life. All organisms are made of, that is basically the structural organization is of tissue, cells, and each cell constitutes what many cells aggregate to form tissues, and tissues organize to form organs, and organs constitute different organs constitute an organ system, and organ system in higher organism they are a perfect organism which consists of many organ systems. So, which is the basic structural and functional unit of life? That means all living organisms are made up of cells. So, what will be the answer? Cell is the basic structural and functional unit of life. Okay, yes. I will tell you one thing. When I am discussing the answer questions, you can do one thing. Take your note. I prefer you to take 100 pages note for this subject. Okay, so that when I am discussing, that is, for example, what was the first question? The first living cell was discovered by. So you can write the answer in your notebook, and after when the answer is appearing, you can check whether the answer is correct or not. Okay, like that. I suggest you to do like that, or simply looking into the screen and listening to me, it won't work very well. Okay, so once the question appears, you can write the answer. It is practical in the case of objective type questions, fill up questions and all. MCQs it will be easy, but in the descriptive questions, either you can copy down the questions or you can you can tell with me, you can follow me, and you can understand how to make up that question. Is that clear to you? Yes. So first two questions I have done for you. And next question, take the odd one out. Amoeba, bacteria, plant, and honeybee. So you can check what is the peculiarity of each one. Amoeba, it is a unicellular organism, and bacteria is also unicellular organism. Plant is a multicellular organism. Okay, organisms can be divided into unicellular, a single celled, and many celled, that is multicellular. So amoeba, bacteria, plant, multicellular, and honeybee, it is also a multicellular organism. So it won't work out because we cannot categorize them as unicellular and multicellular. We have to pick a one and only one odd one. Okay, so you can check in other way, that is whether it is a, how we can again divide organisms, prokaryotic and eukaryote. You can check whether it will work. Amoeba is a prokaryote or eukaryote? Amoeba is a eukaryotic organism. Okay, it having a true nucleus and cytoplasm. So amoeba is a eukaryote, but bacteria is a what organism? Yesterday, in the last session we have discussed what is prokaryote organism. The typical example is bacteria. I have shown you the figure also. Okay, so bacteria is a prokaryotic organism, and plant is a eukaryotic organism, eukaryote, and honeybee is an insect. It is also a eukaryote organism. So what is the odd one here? So these organisms are classified on the basis of true nucleus present. So the odd one here is bacteria. Why bacteria is odd one? Because amoeba, plant, and honeybee all are eukaryotes. So bacteria is the odd one. Bacteria is having a cell wall, and it is having also a nucleoid is present, and it is having a single chromosome which is immersed in the cytoplasm. No other membrane-bound organelles are present. Only organelle present is ribosome. Okay. Then the concept hominis cellula e cellula was put forward by. So what do you mean by this hominis cellula e cellula? Hominis cellula e cellula is a Latin word. Okay, all these are a Latin word. So Latin words hominis cellula e cellula is the meaning of this Latin in English is all new cells exist. All new cells derive from the old cells. Otherwise, we can say that is all existing cells derive from the pre-existing cell. Every cell derived from its old cell. That is the meaning of this hominis cellula e cellula. It was put forward by. So this is concept of one of the concepts of what? Cell theory. So what is cell theory? Cell theory, it has three postulates. What are they? Every organisms are made up of cells, and cells are the structural and fundamental unit of life. And third point was put forward by whom? Rudolf Virchow, and he told that all existing cells arise from the pre-existing cells. The simple meaning is all the new cells arise from the old cells. So this concept hominis cellula e cellula, which means all cells derived from the existing cells, pre-existing cells, was put forward by whom? Rudolf Virchow. The third postulate was put forward by Rudolf Virchow, and other two ones are put forward by Schleiden and Schwann. Okay, so did you notice the answer? So it is Rudolf Virchow. The concept put forward by Rudolf Virchow is hominis cellula e cellula. Then next question, the term protoplasm was coined by. Who coined protoplasm. What is protoplasm is? Protoplasm is the living substance inside a cell. It constitutes all the cytoplasm and all the cell organelles and the nucleus. All these together constitute the protoplasm. So the term protoplasm was coined by whom? In the last session, we have discussed the scientist and its contributions. Okay, the contributions of scientists. In that tabular column, there was a point that is protoplasm was coined by whom? Yes, it was Purkinje who coined the term that is protoplasm. He gave the name protoplasm. So what is the answer? Purkinje. Purkinje is the scientist, name of the scientist. Which is the smallest cell? So the size of the cell may vary. Okay, it may be big or it may be small. So the smallest cell ever known is of a bacteria known as mycoplasma. It is a prokaryote. Okay, so what is the peculiarity of mycoplasma? Mostly bacteria do have a cell wall. Okay, they have a rigid cell wall. But for mycoplasmas, they lack a cell wall. Okay, and also they can survive without oxygen. Can we survive without oxygen? Without oxygen, life is not possible. Okay, so for these organisms, they doesn't have a cell wall, and also they can survive without oxygen, and as a result of this, they can be pathogenic. They act as they are pathogens in plants and animals. Okay, because of the fact that they are resistant to antibiotics, because antibiotics doesn't work on the destruction of cell wall synthesis. Okay, yes. So the smallest cell ever known is mycoplasma. Okay, it's a prokaryote, it's a group of bacteria. Then coming to next question number 7, largest cell is. So which is the largest? The smallest cell is mycoplasma. So which is the largest cell? The largest cell ever known, or the largest of all egg, is the egg, that is ostrich egg. It is the egg of an ostrich is the largest cell ever known, and it is the largest egg. And what is the size? The average size of an ostrich is found to be, it is of a 13 centimeter in length and in width, 13 centimeter width, and an average of 15 centimeter length, and it weighs about 1.5 kgs, which is 20 times bigger than that of a normal chicken egg. Okay, so the size is 20 times bigger than that of a normal egg. So the largest cell ever known is the egg of the ostrich. Okay, it's correct. Then this lactobacillus is dash shaped. So depending upon the shape, bacteria can be divided into 4 classes. That is, spherical shaped bacteria are called cocci, for example staphylococci, and rod shaped bacteria are there, and they are known as bacillus, example lactobacillus. And there are spiral shaped bacteria, and example is treponema. And examples for comma shaped bacteria is a Vibrio cholerae, and they are the group of bacteria known as Vibrio. So lactobacillus comes under which shaped? That is, lactobacillus is a rod shaped bacteria. Okay, so that is the answer. It is rod shaped. Then coming to question number 9, mycoplasma lack dash. Mycoplasma lack, yes, we have just told in the question number 6. Okay, like mycoplasma lack a cell wall. Yes. Then question number 10, phosphate granules are examples for dash. So in the bacteria, there are some structures known as what? Yes, we have discussed in the previous session. That is, there are some bodies known as inclusion bodies, which act as a storage for what? Carbohydrates. Which act as a storage for reserve materials. And for example, phosphate granules are stored in this inclusion bodies which are scattered in the cytoplasm. So phosphate granules are examples for what? Inclusion bodies which are commonly seen in the bacteria. Is that clear? Inclusion bodies. Then, so next group of questions, that is, it is also to test your basics. Directions for questions, that is question 11 to 20. Select the alternative form from the given choices. So they are a group of MCQ questions, multiple choice questions. So we have to pick out the correct option. Okay. So what is it? 11th question. Site for protein manufacture is. What is the site for protein manufacture in a cell? We can read the options. Option A, cell wall. B, cytoplasm. C, ribosome. D, inclusion bodies. So we can start with inclusion bodies because in the previous question we have discussed about inclusion bodies. So site for protein manufacture is not inclusion bodies. It is a storage part. It's a storage for reserve materials like carbohydrates. And in the what about the cell wall? Cell wall, it is not the site for protein manufacture. It is the outer, that is non-living, rigid outer covering of a prokaryotic cell, and also in a plant cell it is present. So it is not the site for protein synthesis. They are meant for maintaining the integrity or the stability of a cell. Then what about cytoplasm? Cytoplasm is the fluid content of the cell in which many cell organelles are scattered, dispersed. So they are also not the site for protein manufacture. Then what about C, ribosome? What is the role of ribosome? Ribosomes are non-membrane bound spherical bodies scattered in the cytoplasm which can be seen both in the prokaryotic and eukaryotic organism. So their function is protein synthesis. They can be seen freely in the cytoplasm, and most probably they are attached to the endoplasmic reticulum, that is RER, rough endoplasmic reticulum. So what is the correct answer? Site for protein manufacture is, what is? It is option C. Ribosomes are the site for protein manufacture. I hope it's clear. Okay, so choice C. Then moving on to next question, that is question number 12. That is, the longest cell is A, mycoplasma, B, neuron, C, PPLO, D is ostrich egg. So longest cell we have to find out. You know the answer, which is the longest cell. Okay, we have studied which is smallest cell, which is the largest cell, and which is the longest cell also. So we can check the options. A, mycoplasma. Mycoplasma, they are the smallest cell. It is not the longest cell. And what about this neuron? B. Yes, neurons are the nerve cell, and we can, when you see the structure, we can see that they are the longest cell with a cell body and many branches extending from it, dendrite, dendrons, axon, axonites, like that. So neurons are the longest cell in our body. So what about this PPLO? PPLOs are pleuropneumonia-like organisms. They are mycoplasma itself. Okay, so it is not the option because pleuropneumonia-like organisms are seen inside what is a category of bacteria. It is not the longest cell. And ostrich egg. What about ostrich egg? Ostrich egg is the what is the largest cell? Okay, so what is the answer? Very easy. Longest cell is neuron. Then, which of the following can change its shape? First one, amoeba. Second, neuron. Third, WBC, that is white blood cells. And fourth one, RBC. So, which of the following can change its shape? You know, first one, when you check the option, first one, Amoeba. Yes, amoeba is a unicellular organism and it is a eukaryotic also, and it can change its shape according to the needs of their body. Okay, so they can walk with pseudopodia like organism. When they happen to see a food, it can engulf the food and forming a wood vacuole, and they can absorb into the body. So amoeba is an example for organism which can change its shape by the streaming movements of its cytoplasm. Then what about neuron? Whether a neuron can change its shape? No, its change is fixed. It is the longest cell in our body. And what about WBC? In the last session, we also told about WBC, that is, it is the white blood cells. They are also known as leukocytes. In our blood, there are different components like white blood cells, red blood cells, and platelets, plasma, like that. So, WBC, what is the function of WBC? That is, WBC white blood cells, they are meant for phagocytosis of the germs or foreign bodies which are happen to enter our body. They are mainly meant for what? Immunity of our body. So whenever foreign organisms enter our body, what will happen is that, like an amoeba, it can change its shape. Its change is flexible, so it can phagocytose. It can engulf that particular particle or organisms which are many of bacteria or any pathogens, and then can destroy that. Okay, so WBC, like amoeba, they doesn't have a fixed shape. Their shape also may vary. Okay, then what about RBC? RBC, the function of RBC is the transport of oxygen. That is, RBC, they are carrying hemoglobin which are meant for the transport of oxygen. Okay, so what is the correct options? 1 and 3. We can check whether any option is like that. A, 1 only. B, 1 and 2. C, 1 and 3. D, 1, 2, 4. No. So what is the thing? What is the answer? 1 and 3. Amoeba and WBC can change its shape. So this is the solution. Solution C. Then moving on to next question. Question number 14. Nucleoid is the region where dash is or are dash are located. So option A, genetic material, B, proteins, C, pigments, and D, nutrients. So nucleoid is a region which is seen in the prokaryotic organisms like bacteria. Why they are known as nucleoid? Because they are not a true nucleus which doesn't bound by a nuclear membrane. The genetic material or the DNA, it is simply scattered in the, it is simply placed in the cytoplasm. There are single chromosome is also present. So nucleoid is the region where what is present, whether it is nucleus or whether it is a nucleoid region, inside it what will be present? The genetic material in the form of DNA will be present. So what is the answer? It is not the proteins, it is not the pigments, it is not the nutrients which is present in the nucleoid region. The genetic material is located. Okay, yes. So solution will be choice A only. Then question number 15. Bacterial cell wall is made up of. So bacteria has a cell wall. So what is the peculiarity of cell wall? So it is a rigid non-living substances. Okay, and outermost boundary. For options we can check A, cellulose, B, peptidoglycan, C, hemicellulose, and D, and it is pectin. So bacterial cell wall is made up of what? If you know the correct answer, we can directly go for the answer. Bacterial cell wall is made up of what? Peptidoglycan. Okay, so for others who couldn't make out the answer properly, I will help you. That is option A, cellulose. Cell wall of a plant cell is made up of cellulose. Okay, cell wall of a plant is made of cellulose. Cellulose is a polymer which is made up of monomeric units like glucose unit. Many glucose units join together to form a polysaccharide known as cellulose. And hemicellulose is a small branched and it is made up of different type of monosaccharides like glucose, galactose, like that. And pectin is a substance which middle lamella is made of. Okay, it is also made of, that is carbohydrate, that is pectin. So here the correct option is peptidoglycans. So bacterial cell wall is made up of peptidoglycans. Okay, yes. We can check the solution is correct. That is choice B. Bacterial cell wall is made of peptidoglycans. Then question number 16. Match the following. Inclusion bodies, that is A, B, mesosome, C, ribosome, D, Golgi complex. So, inclusion borders, it is given, that is eukaryotic, protein manufacturer, cell wall formation for nutrient reservoir. So, we have to check which all can we match together. Inclusion bodies. So you know what inclusion bodies, it is seen in the bacteria. I am not repeating because we have already discussed in the previous question. Its function is storage, or they are meant for the reservoir of food material, excess food materials, or we can tell it as they are nutrient reservoir in the form of carbohydrates or proteins, like that. So inclusion bodies, when we check, cannot be matched with eukaryote, not with protein manufacture, not with cell wall formation or nutrient reservoir. Yes, so it can match with only with nutrient reservoir. So inclusion bodies are nutrient reservoir. Okay, they are placed in the cytoplasm. Then what about mesosomes? Mesosomes are seen in the bacteria. Normally in the bacteria prokaryotic cell, they are the mere invaginations, infoldings of what? Cell membrane. So what is their function? Their functions is cell wall formation, and they also increases the surface area for the respiration to take place. That all things we have studied. So mesosome, they are meant for cell wall formation. So A will match with 4, B with 3. Then ribosome. Ribosome, they are what? They are meant for protein manufacture, for the cytoprotein synthesis. So C2 match with 2. What about Golgi complex? The remaining one is, that is eukaryotic. Why it is eukaryotic? Because Golgi complex is a membrane-bound cell organelle which is seen only in the eukaryotic cell. So, what is the answer? D will match with 1. So, our order will be like this: 4, 3, 2, 1. Like this, isn't it? Yes. So, we can check which option contains that order. It's not A, it's not B, it's not C, it is D, which is the correct option, having the order 4, 3, 2, 1. Isn't it? Is that clear? So, solution D is the correct option. Then, question number 17, select the wrong statement. So, we have to select the wrong statements. You have to read the statements carefully. Then only you can make out which statement is wrong or right. A. Cell walls are surrounded by living, rigid outer layer called cell wall. Please read the sentence. Plant cells are surrounded by living, rigid outer layer called cell wall. This second part is correct. They are made up of rigid outer layer called cell wall, plant cells. But is this sentence fully correct? Not. Which is the wrong word here? Yes, this is. The plant cells are surrounded by living. They are not living. That is plant cells. The cell walls are made up of cellulose, which is a cellulose is a non-living substances. Okay. So that is the thing. This is the wrong statement. Then what about B? Plant cell wall is made up of cellulose. Obviously, they are made of cellulose. Bacterial cell wall is made up of peptidoglycan. Yes, in the last question we have told, find out the answer. That is bacterial cell wall is made up of peptidoglycan. Then D. Both prokaryotic and eukaryotic plasma membrane is composed of lipids and proteins. So, both prokaryotic and eukaryotic, the common fundamental structure or feature is the cell membrane or plasma membrane, isn't it? Yes. So, they are made up of lipids and protein. Last session we have seen the structure of a cell wall, that is it have a two bilipid layer. In between it has proteins are present. So they are made up of lipids and proteins. So the first statement is wrong because plant cells are non-living. They are dead. Okay. So choice A. Then moving on to question number 18. Adjacent plant cells are joined together by A. Middle lamella, B. Plasmodesmata, C. Mesosome, and D. Plasma membrane. So the plant cells, when you look the structure of a tissue or plant cell, you can see that is plant cells are arranged like what? Like the tiles on your floor. Okay, so you can see for fixing a tile, what you have to do? Yes, you have to fix a thing in between, then only it will fix properly. You can use a jelly like substance or cementing like substances. So that is the thing, that is adjacent plant cells are joined together by a fluid like or jelly like cementing substance known as middle lamellae. Okay, middle lamellae, they are the cementing substance in between two cells. Then so we have already find out the answer, but we have to check what all other options are. What is plasmodesmata? Have you heard about plasmodesmata? Plasmodesmata is nothing but they are the channels in between two cells through this middle lamellae. That is, they are the connecting link between the junctions, in between the junctions of the cell. They are the channels which allows the entry and exit of the materials from one cell to another. They are small passages or gateways, we can say. So plasmodesmata are not joined. The option is not correct. The option is middle lamellae. Then what about mesosome? So they are not, because in plant cells mesosomes cannot be seen. Probably mesosomes can seen only in the prokaryotic bacteria-like cell. They are the invaginations of the cell wall. Okay. Then what about plasma membrane? Yes, plasma membrane is seen inner to the cell wall in a plant cell. So they are not correct option. What is the correct option? Yes, correct option is the middle lamella. Adjacent plant cells are joined together by middle lamella. They act as a cementing substance between two cells. Then moving on to question number 19. Which of the following is the correct statement? We have to find out the correct statement. So many of the sentences will be wrong. A. Endoplasmic reticulum is the site of protein manufacture in bacteria. Endoplasmic reticulum is the site of protein manufacture. Whether the sentence is correct? No. Why? Because it is not endoplasmic reticulum, it is a ribosome which is the site of protein manufacture in bacteria. Okay, so in bacteria that is also wrong, because endoplasmic reticulum is not found in bacteria. Endoplasmic reticulum is a membrane-bound cell organelle which can be seen only in the eukaryotic organism. So the sentence is altogether wrong. Endoplasmic reticulum is not the site of protein manufacture, and it cannot be seen in bacteria. In bacteria, ribosomes are the protein manufacturing bacteria. This sentence to be corrected like that. So this sentence is wrong. Then B. The membrane-bound inclusion bodies help in the storage of nutrients in prokaryote. So this sentence is wrong because inclusion bodies can be seen in the prokaryotes. The function is also the storage of nutrients, but what is here written as the membrane-bound inclusion bodies. So you know what is the peculiarity of prokaryotes? What's the main difference? In prokaryotes, no membrane-bound organelles or no membrane-bound bodies could be seen. So membrane-bound inclusion bodies, that statement is also wrong. C. The living material inside the cell is called protoplasm. Yes, the living material inside the cell is protoplasm. That is correct. A cell will be having a cell wall, or if it is a plant cell, have a cell wall. Other than it has a cell membrane or plasma membrane. Inner to that, it has the living material inside. That is, what all things? Nucleus will be there, cytoplasm will be there, and cell organelles will be there. All the things constitute the protoplasm. So that statement is true. Then D. Mesosome is the cell wall extension which helps in photosynthesis. Mesosome is a cell wall extension. That part is correct. It is a cell wall extension which can be seen in the prokaryote like bacteria. Then it is said that which helps in photosynthesis. No, because bacteria, many of the bacteria, they could not perform photosynthesis, even though photosynthesis is carrying out in some bacteria like cyanobacteria, and it is not true bacteria at all. Okay, and which helps in photosynthesis, that statement is wrong because mesosome is cell wall extension which helps in the respiration. Okay, so that sentence is also wrong. Is that clear? Yes. So which is the wrong statement? That is choice C. Is that clear? Yes. Then 20. Which one of the following are included in protoplasm? What is that? Which one of the following are included in protoplasm? First one, nucleus. Second one, cytoplasm. Third one, organelles. And fourth one, nucleoplasm. So what all things are included in the protoplasm? Protoplasm is the living substance of a cell which includes nucleus is present, cytoplasm is present, third cell organelle is present, and nucleoplasm. What do you mean by nucleoplasm? When you will study the structure of a nucleus, you will be able to see a nucleus. The outer membrane is nuclear membrane. In between the membrane, there are pores known as nuclear pores. Inside it is thread-like genetic material called chromatin material is present. And inside the nucleus, the chromatin material is dispersed or scattered in a fluid-like substance which is similar to that of cytoplasm, that is nucleoplasm is present. So nucleoplasm is the fluid-like substance which is present inside the nucleus. So if nucleus is included in protoplasm, probably nucleoplasm will also comes under the protoplasm, isn't it? Because nucleoplasm is situated inside the nucleus. So all the things 1, 2, 3 and 4 will come under the part of protoplasm. So what is the option A? 1 and 3, B 1, 2 and 3, C 2 only. What about D? 1, 2, 3 and 4. So all the things are coming under protoplasm. We can check whether either is correct. Yes, choice D, the correct postulated cell theory. And what are the main postulates? So we can revise the thing that we have studied. That is, cell theory was proposed by Jacob Matthias Schleiden in 1838 and Schwann in 1839. Later it was modified by Rudolf Virchow. These are the points coming under who postulated cell theory. Then what about the second part? What are the main postulates? That is, cell theory consists of three postulates, that is, all living organisms are made of cells, and cells are the basic structural and fundamental unit of life. Then all cells derive from pre-existing cells. So these are the postulates. So let us check whether the assumption is correct or not. Cell theory was proposed by Jacob Matthias Fried in 1838 and Theodor Schwann in 1839. Later it was modified by Rudolf Virchow. The main postulates are all organisms are made of cell. Cell is the basic structural and functional unit of life. All cells arise from pre-existing cells only. That is, omnis cellula e cellulas. All cells arise from pre-existing cells only. Then, next question. What is the next question? How Staphylococcus differ from Lactobacillus in shape? So, what is the shape of Staphylococcus? Cocci. So, Staphylococcus comes under the group of cocci bacteria, which is spherical in shape. Okay. What about Lactobacillus? Lactobacillus, which come under the category of bacilli, bacillus, which is of what shape? Rod shaped. So, Staphylococcus is spherical in shape and Lactobacillus is rod shaped. Is that clear? Yes. Then, moving on to next question, that is question number 23. What are mesosomes? Write their significance in cell. So what are mesosomes? Mesosomes are the mere invaginations of the extensions of the cell wall, which can be seen in the prokaryotic cell. And what is their significance? Yes, their significance, they help in the cell wall formation and also in the respiration, because it increases the surface area of a cell wall where the respiration can take place. What is respiration? It is the process of breaking down of food to release energy in the presence of oxygen, probably in normal cells. So we can check whether the points are correct or not. Mesosomes are structures seen in prokaryotes which are formed by plasma membrane extensions into the cell. Mesosomes helps in cellular activities such as cell wall formation and respiration. Okay. Then next type is understanding based questions. So the question coming under understanding based question is, it is the question to check your understanding level, whether you have understood the concept. Question number 24. Differentiate between prokaryotic cell and eukaryotic cell. Yes, it is very easy. So what is the answer? Prokaryotic cell, the size may vary between a prokaryotic and eukaryotic cell, and in a prokaryotic cell, nucleoid is present. In eukaryotic cell, that is a true nucleus is present. In prokaryotic cell, membrane-bound cell organelles are absent. In eukaryotic, membrane-bound cell organelle is present. In prokaryote, only single chromosome is present. Eukaryotic, many chromosomes are present. Okay, so most probably the size of the prokaryotic organism is very small, that is 1 micrometer in length, and size of eukaryotic cell will be little small, 5 to 10 micrometer. Okay, so we can check. Yes, size generally 1 to 10 micrometer, size generally under eukaryotic 5 to 100 micrometer, and nucleus is absent, that is nucleus is present, contains single chromosome, more than one chromosome, membrane-bound cell organelles like endoplasmic reticulum, Golgi apparatus, etc. is present in the case of prokaryotic cell. In eukaryotic cell, membrane-bound cell organelles are present. So these are the difference between a prokaryotic cell and a eukaryotic cell. Is that clear? Question. That means to check whether you have understood the concept. Okay. So 25th question. Unlike prokaryote, all eukaryotic cells are not identical. Justify the statement. What is the meaning of this question? That is, how the eukaryotic cells, most of the prokaryotic cells, there is a difference between prokaryotic and eukaryotic cells. But among the prokaryotic cells, there is no prominent differences because almost all the prokaryotic cells like bacteria are same. They have a cell wall, they have a cell membrane, they have ribosomes, and they have chromatin material or genetic material present. So there is no much differences in their structure of prokaryote. But in the case of eukaryotic, for example, eukaryotic plant cells are eukaryotic cell and animal cells are eukaryotic cell. Eukaryotes are included under protista, fungi, plantae, and animals. But in the case of plant cell, a cell wall is present. But in animal cell, a cell wall is absent, only cell membrane or plasma membrane present. And also there is another difference also, that is plastids, that is color containing compounds or storage compounds are not cell organ. They are cell organelles. Plastids are not present in the animal cell. Plastids are present in the plant cell. Then in the case of plant cell, a large vacuole is present which acts as the storage organ of plant cell. But in the case of animal cell, not much large or predominant vacuoles are not present. If it is present, they have only a small size. Okay, so that is the answer. So we can check. Eukaryotic include protista, plants, animals, fungi, etc. So these all are kingdoms of eukaryote. All of them show considerable difference in their structure and organization. Yes, for example, plant cells are surrounded by an outer cell wall, whereas cell wall is present in animal cell. Similarly, plastids and large central vacuole are present in plants but absent in animals. And if vacuole is present, it will be small. So this is the difference between the plant cell and animal cell. But the common feature is that both are eukaryotic cells because they have a prominent or well developed or true nucleus is present. Okay. Then moving on to question number 26. Why plasma membrane is said to be semi-permeable membrane? Yes, the important point is plasma membrane is said to be semi-permeable. They are permeable, they are not fully permeable. The meaning of the word semi means half. Half permeable means it will not allow all the materials to pass in and out of the cell. It will restrict the entry of some of the molecules. That is, it allow only particular substance to move in and out of the cell, or they are like watchman or gateman, that is, who is restricting the entry and exit of some of the persons. Okay, so they are known as semi-permeable membrane. Okay. Then, plasma membrane is made of proteins and lipids. They are said to be semi-permeable membrane as they allow the entry and exit of only some of the materials and they will prevent some others. Okay, depending upon the need of the cell. Then, there are another last group of question, that is application based question. This question is based on your application level of knowledge, whether you can apply the concepts that you have studied in your basics. So question number 27, complete the table given below. So we have to check whether we can do it or not. So this is the structure coming under this structure, and these are the features. So A feature is given. Okay, plasma membrane extension. So we have to find out which structure is doing the function or feature, that is plasma membrane extension. So what is who is doing this plasma membrane extension? Yes, plasma membrane extension is the function of what? mesosomes, which is seen in the prokaryotic cell. So, which are helping the cell wall synthesis and also in the respiration. So, plasma membrane extensions are the function or feature of a mesosome, which increases the surface area of the plasma membrane where the respiration is taking place. Okay. So, mesosomes are the plasma membrane extensions. Then B, ribosome. They are site of dash. Very easy question, isn't it? They are site of what? They are site of protein synthesis. What is the peculiarity of ribosome? Yes, ribosomes are non-membrane bound cell organelles which can be seen both in prokaryotic cell and the eukaryotic cell. So, in prokaryotic cell like bacteria, they are simply immersed in the cytoplasm. In eukaryotic cell, these ribosomes are seen normally attached to endoplasmic reticulum, hence it got its name rough endoplasmic reticulum if ribosomes are attached. What is their function? Ribosomes can also be seen freely in the cytoplasm of the cell. So, the function of ribosome is protein synthesis. Okay. Protein synthesis or protein manufacture. You can use that word also. Then, feature is given, joins adjacent plant cells. So, a plant cell which is the cementing substance or which is the fluid substance or jelly-like substance which cements or which joins to adjacent plant cells. Yes, what is that? The name? That is middle lamella. Middle lamella means from the name itself you can recognize its function. That is, it is present in the middle or in between two cells. Okay, so it is middle lamella which joins adjacent plant cells. Then, yes, our assumption is correct. Then D, plasma membrane. Plasma membrane made of dash and dash. So, plasma membrane is a cell wall is made up of cellulose in plant cell. A cell wall is made up of peptidoglycan in plant cells. And plasma membrane, they are made of what? Lipids and proteins. They have a bilipid layer. Their outer surface is made up of two lipid layers, and in between these lipid layers, what is present? Proteins are present. Different kinds of proteins are present. So we can check, that is plasma membrane. They are made of lipids and proteins. Lipid means they are fats like oils, fats, animal fats like that. E, structure is not given, feature is given. Living material inside the cell. So, inside the cell, cell organelle is present, nucleus is present, cytoplasm is present, and it is gets surrounded by cell wall and cell membrane, or either only cell membrane in animal cell. So, the living material found inside is apart from cell wall and cell membrane. We cannot include that because they are forming the outer wall of the cell. Living material inside it includes the protoplasm, which consists of cytoplasm, nucleus, and all the cell organelles like endoplasmic reticulum, Golgi bodies, ribosomes like that. Okay. Yes, protoplasm is the living material inside the cell. Then next question F. Fluid content inside the cell membrane. So inside the cell membrane, the fluid content. Apart from this, cell organelles and nucleus, we have a nucleus content. Fluid content. Sorry, not nucleus content, fluid content. That is, what is the fluid content? It is the cytoplasm. So we can make a formula like this. Protoplasm is equal to cytoplasm plus cell organelles plus nucleus. So therefore, cytoplasm is what? Yes, all the thing that is inside the protoplasm, the nucleus and the cell organelles, we can minus nucleus and cell organelles, we will get cytoplasm. Okay. When we are removing the cell organelles and nucleus, the only fluid content, that part is known as cytoplasm. So, cytoplasm is the fluid content inside the cell membrane. So these all are the questions that we have discussed so far, and I think the basics one, the first part of that is the first part of unit, the fundamental, I mean the basic unit of life, that is cell, the basic unit of life. It was thorough for you and it will be helpful for you in your as a foundation course. It will be helpful for you in your future studies. And in the next session we will be continuing our basics part 2. In that we'll be discussing the cell organelles and many other things. So I wish you all the very success and having a wonderful day.